So what we're going to do today is we're going to start to put things in motion. We're going to start off with a little reminder where we left off last time about the organization of our thick and thin filaments. What's the last name of our thick filament? Thick myosin and thin actin. So if we look in the upper right-hand corner, this is our typical picture that we saw last time at the end of class. But over on the right, hopefully you can relate this transmission electron microscopic picture of an uncontracted sarcomere. And what's a sarcomere from where to where? Z to Z. Z squared is a sarcomere, Z to Z. And so we can see our Z lines very easily in our electron microscopic picture. And remember, that's right in the middle of the I bands. You see how the I bands are lighter than the A bands? And then over on the left-hand side, we see the I bands are light because they only are comprised of the actin thin filaments. But notice, it's not all of the actin. Some of the actin extends into the first part of the A band. And we can see the A band is going to extend from the end of the thick filament to the opposite end of the thick filament. And we have some of that overlapping region that's going to make for the darkest part of our A band. It's dark because the middle of the A band is lighter. You see the H zone? That H zone is where the actin thin filaments have stopped. And what you only have remaining there are going to be the thick myosin. So there's less stuff in the middle of the A than you see on the edges. And then finally, right in the middle, you have the M line. Those are the structural proteins that are going to hold those thick filaments in the right position and right orientation to get the sliding action. Does that picture help a little bit when you look at the lighter bands and the darker bands? Sometimes being able to see this helps understand the little illustration, and sometimes the illustration helps with understanding why it's light and why it's dark. So that's why I wanted to start off with that. So now let's start the process of having a contraction. We're going to look at skeletal muscle. Everything we're talking about today is really referring to skeletal muscle. And with skeletal muscle, they will not spontaneously contract. You have to have a signal from a nerve for a skeletal muscle to contract. Now, does that mean all skeletal muscles are voluntary? Are all skeletal muscles voluntarily controlled? In fact, the answer is yes and kind of no. You can voluntarily control all of your skeletal muscle. The other question, though, is are there some skeletal muscles that can also be involuntarily controlled? Absolutely. Because your blinking of your eyelids, that's skeletal muscle. It's voluntarily controlled, right? You can just close them and keep them closed. Some of you do that every day. Not calling anybody's name, weekend. No, he stays awake. Your diaphragm. Your diaphragm is skeletal muscle. Sometimes this is where I insert a blonde joke. There are too many blondes. I'm not going to do that. You don't have to think breathe in, breathe out, right? It's subconsciously controlled. But you can control it, right? You can hold your breath. But if you hold it long enough and pass out, what are you going to start doing? You're going to start breathing again. But the bottom line is we have to have a signal to the muscle for it to contract. That signal comes from our nervous system. And we have the central nervous system, which is the brain and the spinal cord. And from the spinal cord, we have peripheral nerves that extend away out to the body. And the nerves that send signals to muscles or to glands to cause something to happen are called motor nerves. And a nerve cell is called a neuron. Now, nerves are the next chapter. We're going to get to it. So this is a little bit of a preview, and they're sort of tied together here. Now, notice as our motor nerves, as they approach the muscle cells. Now we've got a number of muscle cells here. These aren't myofibrils. Each one of these is a muscle fiber. And what's another name for a muscle fiber? A muscle cell. Muscle fiber is a muscle cell. But notice as the motor nerve approaches the muscle, it branches. And several collaterals will contact and excite multiple fibers. Now, some of these motor nerves will excite 2, 3, 5 fibers in some muscles. Some will excite 100 or 1,000, depending on the muscle and what that muscle is used for. Your quadriceps muscle in your leg, it's all about power. And so one motor neuron is going to cause 1,000 muscle fibers to contract because you're going to have to do a lot of work. The ciliary muscle that controls the shape of the lens of your eye, one motor nerve may cause 10 fibers to contract because you're doing a lot more delicate work with that. But the motor neuron, the motor nerve, and the muscle cells or fibers it's attached to is referred to as a motor unit. Now, this is a freebie. We talk about this a little bit more in depth in our majors physiology class, which is for juniors, not for freshmen. But when we look at motor units, your body has adapted and evolved to do two things. One is to maintain homeostasis. Have we sort of hammered homeostasis to death already in this class? The balance of your body. Your body's evolved to maintain homeostasis. The other thing that your body has done and evolved to do is to conserve energy. You're going to conserve energy at all cost. And so when you look at motor units, there is a principle called Henman's size principle. You don't have to write this down. Again, like I said, this is a freebie. Henman's size principle deals with the number of muscle fibers needed to accomplish work. Because I think you can already understand if your muscles are going to contract, you've got to use energy. So if I am going to do an exercise, do a curl, all right, am I going to have to work as hard to curl the empty bar, which is 45 pounds, or is it going to be harder for me to curl 200 pounds? Well, that's a no-brainer, right? Heavier weight, it's going to require more work. Am I going to engage the same number of muscle fibers to curl 45 pounds as I do 200? No. I mean, if I contracted the muscle hard enough to lift 200 pounds, if I contracted that many fibers with just a bar, I would slam myself in the head. Boom. That's never going to happen. I don't work that hard in the first place. So Henman's size principle says as soon as your muscles are signaled to work, you are going to stimulate the fewest number needed to accomplish the task. And so you're going to start off, the little, central nervous system conductor is going to say, motor unit 1, go. Motor unit 1 fires. I'm not lifting the weight. Little guy's going to say, motor unit 2, engage. The second one's going to join in, try to help. The guy says, okay, 3, help. We need help. And you're going to continue to engage motor units until you accomplish the task. Now, let's say I'm going to try to curl 600 pounds. If I can get it off the floor even, am I going to curl 600 pounds? No, but my brain is going to engage every single motor unit I have until my brain says, okay, you're stupid, put it down, or stop trying to pick it up, or my muscles completely fatigue and they force me to stop. So do you see how that works? Because if I'm going to curl this remote, I don't have to use a lot of energy. I don't want to use all my muscles. So that's a way that your body conserves energy. So the arrangement of these motor units, the number of fibers that are actually excited to contract, again, it's a way to conserve energy. Now, we're approaching this topic of how do we signal a muscle to contract. And so at the end of our motor neuron, at the end of our nerve cell, we have this enlarged end sometimes called a button. I think your book calls it a knob. It's this enlarged part at the end. Where we have this end of our nerve cell and we have the sarcolemma of the muscle beneath with a little short space between. That's referred to as a neuromuscular junction. Sometimes you may hear it referred to as a myoneural junction. Either way, you're talking about a nerve, you're talking about a muscle together. And this is going to be the place where we signal from the nerve to the muscle, you need to contract. Now, this type of junction is referred to as a synapse. We often refer to these because of the neurons and the nerve cells. And in my science major, junior physiology class, I always go over the nervous system first, because it's important to understand how the signals are transducted down the neuron before we get to, okay, it being transferred to the muscle and repropagated in the muscle membrane itself. Here in A&P, they've arranged it backwards, and I wanted to stick with lab and stick what people have done in the past. I didn't have long to get going this semester. So we're kind of approaching it a little bit backwards. So we're going to talk about a lot about the electrical signal today. And just understand, it will relate to the nerve when we get to that part. But when we have this type of junction, where we're going to take the electrical signal that is sliding down our nerve, and we need to transfer that electrical signal to the muscle sarcolemma and regenerate an electrical signal in that membrane, we're going to transduce, we're going to change the signal from an electrical into a chemical signal that we regenerate into an electrical signal. That change from electrical to electrical is the synapse. And you see that between neurons, neuron to neuron, you see it neuron to muscle, you see it neuron to gland. And so we're going to focus the first part of our discussion about signaling a muscle to contract is all going to be about this synapse. And again, with the synapse, it's going to have three things. It's going to have the end of the neuron, which is the knob, the terminal knob, the synaptic knob, the terminal button, whatever you want to call it. The membrane of the muscle underneath is the motor end plate. And the small space between those two is called the synaptic cleft. That's our synapse. Those are the players of our synapse. Now, let's focus on the end of the neuron here. The knob, the swollen portion at the end. Within this end of the axon, we're going to have a lot of vesicles. And I believe we're going to have on the order of about 10,000 vesicles. That's a lot, right? 10,000 vesicles at the end of our axon. And these vesicles are filled with a chemical. Now, for skeletal muscles, to stimulate and excite a skeletal muscle, because that's all we're going to do. We're going to cause it to contract or not contract. So we don't have to have an off switch. We just need the on switch, because the default for a skeletal muscle is to be relaxed. So our chemical that is going to excite the skeletal muscle is and always will be acetylcholine. That is a chemical called a neurotransmitter. Neuro because it's produced by a nerve. Transmitter because this is the transmitted signal that's going to cross that synaptic cleft from the terminal knob to our motor end plate or our muscle membrane. So we can spell it out, acetylcholine. The abbreviation is ACH. My pre-meds, they have fill-in-the-blank short answer exams. We've just got multiple choice, so you just have to be able to recognize that. You don't have to spell out acetylcholine. But understand, that's what we're going to be using here for skeletal muscle. Now, on the membrane side, we have receptors for acetylcholine. Now, this is where we have to have a little bit deeper understanding of what's going on. Whenever you touch a hot stove, how long does it take for your hand to move away from that stove? Instantly, rapidly. So we need that signal from the nerve to the muscle to happen rapidly. Now, these electrical waves that we're going to talk about, they're called action potentials, and we're going to use that word a lot, and we're going to talk more about the action potential. These action potentials can flow along your nerves at 100 meters per second. That's pretty fast, right? 100 meters per second. I think Usain Bolt's record is 100 meters in 9 something. What's Bolt's world record for 100 meters? 9.8? Yeah. So we're going to kill Usain Bolt. He's barely going to be out of the blocks, and our action potential is already finished. But once we get to our synapse, we have to rapidly chemically signal the muscle. Now, how is this chemical going to be transported from the membrane of the axon to the motor end plate, the sarcolemma? How's it going to be transported? Diffusion. It's simple diffusion. These vesicles are going to undergo exocytosis and release the neurotransmitter. So when we talk about diffusion, how can we increase the rate of diffusion? Create a steeper gradient, right? How many vesicles did I say we had in our terminal knob? 10,000. Within each of those 10,000 vesicles, there are 30,000 molecules of acetylcholine. 10,000 times 30,000 equals a lot. How much acetylcholine is in the synaptic cleft? None. So we're going from 10,000 times 30,000 to 0. Do you see how steep that gradient is? It's skydiving. So that means when we release these neurotransmitters from the end of the nerve, it's going to be a tidal wave of acetylcholine, a tsunami that's going to wash across this space. And that space is only, I believe, about 20 micrometers, if that. It may be 2. I'm remembering a 2. So it's either 2 or 20 microns. It's a short distance. So that tidal wave of acetylcholine is going to wash across and slam into our motor end plate, our sarcolemma. Now, our receptors are in our sarcolemma. And so we need the signal 1 to move rapidly, but we need to detect it rapidly. And if we want to detect the acetylcholine more rapidly, what do we need more of in our muscle membrane? The receptors. And how can we package more receptors into a certain space? Increase your surface area so you can have more receptors. And we do that by folding in the membrane. You see how it's folded there? When we fold in the membrane, we've increased the surface area so we can have more acetylcholine receptors. So we have a tidal wave of signal, and we have an army of receptors to detect the signal. That will speed up the chemical signaling that happens at our synapse. So this is going to be the first part. This is going to be what we term the excitation of our skeletal muscle. Okay, don't panic. This is a big overview picture. I'm a big picture person, and I like to start out with the big picture and then look at the small details. So what we're going to accomplish today and Friday, we don't have a lot of slides because we're going to take our time going through this. But there's a lot of information, a lot of concepts we got to grasp. From the motor nerve to the point at which we contract the muscle, we're going to take it in 3 steps. And our first step that we've already begun to look at is at our neuromuscular junction, and it's called the excitation step. From the excitation step, where we take our electrical signal from the nerve and transduce it into a chemical signal, we're then going to further transduce that chemical signal back into an electrical signal. And that's going to be step 2, the excitation-contraction coupling. And we're going to create that electrical signal that's referred to as an action potential. There's that word again. And that electrical wave is going to wash across our plasma membrane of our muscle, and it's going to wash down the T tubule, where then we release the calcium from the sarcoplasmic reticulum. And that calcium then is going to cause us to go into cross-bridge cycling, which I like to call contraction. So we've got excitation, step one. Contraction is the third step. And the thing that ties the excitation and contraction together, they call the coupling. So excitation, coupling, contraction. Those are our three steps that we're going to go through. And for each one of these, we have a short video to watch. And then we'll walk through the steps in our slides so that we're all on the same page. All right? So, first step, this is going to be at our neuromuscular junction. This is going to be utilizing acetylcholine and the acetylcholine receptors.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma.
Did you see in the nerve, the yellow nerve, how that white wave just kind of flowed along that nerve? That is our action potential. Now, an action potential is simply a flipping of our membrane potential. Now, what number did we say was our resting membrane potential last time? Was it negative or positive? Negative. It was in the neighborhood of negative 90. We said negative 70 was a number that you could hang on to. The action potential is basically a wave of change of voltage where the membrane goes from negative to positive and back to negative again. On a graph, it looks like a spike. And that spike and all of those patches of the membrane with all of these ion channels all along the length of a membrane, that ion change flows down that axon much like the wave at the football game. Remember how we had that analogy? And we're going to see it towards the end of today, but I want you to keep that in mind as we go through these. Yes, ma'am? So cramping is going to be caused typically, when you see folks cramping, like football players cramping, are they going to cramp at the beginning of the game or end of the game? End of the game. Are they going to cramp when it's cold or when it's hot? They're going to cramp when it's hot. Because at the end of the game, when it's hot, what condition are they in? Dehydrated. When you dehydrate, you're going to increase the concentration of your electrolytes, your ions, around the muscle. And as you increase the ions around the muscle, it changes your membrane potential. And if you change your membrane potential enough, your muscle is going to think it's being signaled by a nerve, and it's going to contract. And it's not going to stop. So that's why when you see people with cramps, trainers are massaging the muscle, trying to increase the circulation to get more fluid to the muscle to balance out the ions. The other thing that you're going to have to do is you're going to have to drink, or they'll give you an IV to try to get you hydrated to balance out those ions. Because it's the ions we're going to see that do the signaling to contract. And if you jack them up, cramp. All right? So, our nerve is signaling action potential, washing down our nerve cell. And when it gets to the end, our terminal knob, synaptic knob, we're going to have some voltage-gated channels. Now, these voltage-gated channels are closed until we change our membrane to positive, and that's going to happen when an action potential gets there. And so when we get that action potential, this positive change of our membrane voltage, when it gets to the end, voltage-gated, what ion is that? Calcium channels open. Now, much like sodium, calcium is in high concentration outside the cell. And so when the action potential changes the voltage, voltage-gated calcium channels open. Where does calcium go? It comes in by facilitated diffusion. And calcium starts a signaling cascade. Now, have you ever stacked up dominoes? Then you knock the first one over and it knocks the second and the third and so on. Calcium's the first domino. The very last domino in this whole process is that the calcium will lead to the fusion of the vesicles to the membrane and the exocytosis of acetylcholine into the synaptic cleft. Calcium doesn't directly do it. There are a number of steps involved. We're not going to get into those steps. You just know calcium triggers indirectly the exocytosis of acetylcholine. But what caused calcium to come in? The action potential getting to the end of our nerve cell. Now, we said we have 10,000 vesicles filled with 30,000 molecules. This tidal wave of acetylcholine washes across this very short synaptic cleft, and it slams into the motor end plate where we have our acetylcholine receptors. A lot of molecules move fast. A lot of receptors detect it quickly. And once we bind the acetylcholine to our receptors, now we initiate the second part of this process, the coupling of the excitation with the contraction. So when we bind acetylcholine, that's the excitation part. Because again, skeletal muscles, they're relaxed, they're off when nothing's happening. All you have to do is turn them on. And that's the excitation. Acetylcholine will always excite a skeletal muscle. Any questions about part one before we move on? Signaling from the nerve involves the calcium release because of change in voltage. The calcium leads to the exocytosis of acetylcholine. Acetylcholine crosses our space and it binds to the receptors. We good? All right. Now, here we get into the coupling. Just some reminders before we get there. Our sarcolemma that we see across here, phospholipids. What characteristic do phospholipids have? Think back to our discussion of macromolecules. What was that word that had the molecule that had the two different parts? Okay, part of it is hydrophilic. That's going to be our nonpolar tails. Part is polar. What? Amphipathic. Now, because of the amphipathic nature of our phospholipids, that makes it a semi-permeable membrane, right? And because it's semi-permeable, ions aren't going to pass. And since ions aren't going to pass, we've got it set up so we've got a lot of sodium outside, a lot of potassium inside. Now we've got calcium outside high as well. We can add that to the picture. Not so critical for our muscles, but it was critical for our nerve and signaling. And since we have so much sodium out, and remember we've got all those negative molecules that are locked inside the cell, that means our resting membrane potential is what? Positive or negative? It's negative. The inside's more negative. Now the other thing we need to remember before we get on with this coupling is our sarcolemma dives down into the cell. And what do we call those invaginations? T-tubules. T is for transverse. See how this is about 90 degrees to our plasma membrane? That's why they call them transverse tubules. So we have receptors for acetylcholine directly in our motor end plate. But I want you to notice, just lateral to our motor end plate in every direction, we have different kinds of receptors all along our sarcolemma and all down in our T-tubules. These other channels are going to be critical in this wave of electrical change that's going to flow across our sarcolemma. Oops, I knew there was a video. So here's part 2, the coupling of the excitation with our contraction.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus 2 adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle.
You know what I think is pretty cool? It's when you look at what we've done so far, a nerve signaled the muscle by causing acetylcholine to be released, and calcium did that. I think you can already tell based on what we talked about last time with tropomyosin and troponin, the release of calcium here is going to signal the muscle to contract. So calcium signals the muscle cell to be excited, and calcium is going to signal a contraction to begin. So calcium, it's pretty important, right? All right, so part 2. We're back at our neuromuscular junction. And we're at our acetylcholine receptors. Acetylcholine has been released by exocytosis, and it binds to our receptor. Now, this particular receptor in skeletal muscle requires 2 acetylcholine molecules to bind. If one binds, nothing happens. So this is different from a voltage-gated channel in the sense you've got to have chemicals or you have to have a molecule that binds to a receptor, which is called ligand. So this is either referred to as a chemically gated or a ligand-gated channel. And in the video, they talked about it being a cation channel. The reason is, when you bind two acetylcholine molecules, it opens. And when it opens, it will only allow sodium and a little bit of potassium to diffuse. So again, we're talking facilitated diffusion. So when two acetylcholine bind, sodium goes where? Think about where sodium is high concentration. You open a channel for sodium, where does it go? It flows in, down its concentration gradient. And remember, that gradient is like 150 out and 5 in. It's steep. So it comes in rapidly. And a little bit of potassium goes out. So what was our resting membrane potential, positive or negative? It was negative. So when we allow sodium that's positive to come flooding in, our membrane potential is going to flip from negative to positive instantly. And as that flip happens and sodium comes in, that sodium is going to spread all underneath the membrane in every direction. And those positive sodium ions are going to locally change the membrane potential. You know, you drop a rock in a pond, you see the ripples spreading out. The sodium is going to change that membrane potential like the ripples of the water for a pretty good distance underneath that membrane. And so that positive change in our membrane potential is going to excite another channel that is right beside our neuromuscular junction. And these are those channels we saw in the illustration that are all along the length of our sarcolemma and down into the T-tubules. These are voltage-gated sodium channels. So sodium came in because of our acetylcholine receptor. It changed the local membrane potential underneath the terminal knob, but because sodium flows in every direction, it's also, here you see all this sodium coming in, it's also going to change the potential underneath this first sodium voltage-gated channel. When that sodium voltage-gated channel opens, where's the sodium going to go? It's going to come in because we still have high sodium out. When sodium comes in here, where does it go? Everywhere, like the ripples in a pond. And that sodium then is going to stimulate the next sodium voltage-gated channel beside it to open. When sodium comes into that one, it's like the wave at a football game. One person stands up. When they start to sit down, that's the signal for the next person to stand up, and the next person, and the next person. And that's going to be the change in the electrical wave that washes across the membrane. In altered muscle potential. Sorry. Muscle potential. That's another word for action potential in muscles. Maybe that was on the last slide and I missed it. Sorry. So here is this is the way it kind of works. Okay? So this is our sarcolemma. All right, what's your name? Chelsea. Chelsea is no longer Chelsea. She is a acetylcholine receptor. So when you see her on campus, just say, hey, acetylcholine receptor. So when acetylcholine binds, where does sodium go when this opens? It goes in. It spreads underneath the membrane. What's your name? Allie. Nope. Sodium voltage-gated channel for the rest of the semester. Sorry. What's your name? So the membrane potential changes outward. It's going to cause the sodium voltage-gated channel to reach its threshold, and we're going to talk about that in just a minute. It's going to reach its threshold, and it's going to open. When this sodium voltage-gated channel opens, where does sodium go? In. Spreads out in every direction. What's your name? Jessica. Sodium voltage-gated channel. This voltage-gated channel is going to cause this one to open. Sodium comes in and goes everywhere. Sodium voltage-gated channel. You see what's happening? We're reestablishing the opening in sodium. And as we do that and we flip our negative membrane potential, sodium comes in positive, 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 positive. That wave of change is going to wash all along the length of our sarcolemma and wash down the T-tubule. Does everyone understand how that works? So, that action potential, that muscle potential, membrane potential, MP is membrane potential, I'm sorry. Membrane potential, and this muscle impulse is another term for action potential. That's going to flow all across our membrane. And so what we have done by this step is we've taken the electrical signal from the neuron, changed it into a chemical signal. What is our chemical signaler? Acetylcholine, that neurotransmitter. And now we have transduced it back into an electrical signal that in our skeletal muscle, it can travel at about 1 meter per second. It's still kind of fast. Neuron can do it, insulated neuron can do it 100 meters per second. Muscle is about 1 meter per second. So here's what we're talking about. There's a sodium voltage gated channel, all along the length. But notice there's another voltage gated channel beside it too. It's a potassium voltage gated channel, because what relationship do sodium and potassium have? Opposites. So when we look at this action potential, this change in membrane potential, I want you to look at this graph. So here's resting membrane potential, negative 90 or so. In this number 2, where we see our potential becoming more positive, that is our acetylcholine receptor letting sodium in, making our membrane potential, what our videos say, less negative. It's another way of saying more positive. So we're going upward. And when we reach, I know this says minus 65, but I want the number you to remember to be minus 55. Write that down somewhere. When your membrane potential reaches minus 55 millivolts, that's the threshold to open a sodium voltage gated channel. And when you open a sodium voltage gated channel, you have just restarted the action potential. The only way you can have an action potential is to open a sodium voltage gated channel, period. If you don't have a sodium voltage gated channel, you will not have an action potential. And when we get to nervous system, we're going to see there are parts of our nerve cells that purposefully don't have sodium voltage gated channels. They have other receptors, but not these sodium voltage gated channels. So when in step 2 we let sodium in because of our acetylcholine receptor, we reach minus 55, that opens our sodium voltage gated channel. It opens and closes fairly quickly, but not fast enough that it doesn't flip our membrane potential up to near positive 30. Well, now our potential's reversed, right? We're no longer negative inside, we're positive. And if we ever want to be able to contract again, we need to get it back negative as quickly as possible. And that's this step number 4. You see it coming back down? That's because in step number 4, we open potassium voltage gated channels. Potassium, being highest inside, now goes out. And when you release all those positive ions out, our membrane becomes more negative inside, and it resets the charge difference. So you see how that looks like a wave? And if you take all those sodium and potassium voltage gated channels lined up all across the membrane, that flip and flip back of our membrane potential, it's going to look like a wave flowing across our membrane and down into the T-tubules. That's the action potential when you set it in motion. That's going to be the same thing that happens in your neuron when we get to nervous system, that action potential. I wanted to spend a little more time doing that. And this little animation kind of shows you how the blue lines are our sodium coming in, the pink lines or the red are potassium going out, and you see how that continues to just flow down our membrane. Each one stimulating the next, just like at a football game. You know when to stand up and sit down because of what the person next to you does. That's our action potential. And the last part of this coupling, right before we get to contraction, is when that action potential, that's our AP, when that voltage flipping, when it slides down our T-tubule, this is our T-tubule, there's our sodium voltage gated channel, potassium voltage gated channel. When those get to that point where our T-tubule is in contact with our terminal cisternae, remember that's part of our sarcoplasmic reticulum, that voltage flip is going to twist that voltage gated calcium channel, and that's going to open a calcium release channel in our sarcoplasmic reticulum. And where does calcium glow? Glow? Where does calcium go? It floods into our cytoplasm. It leaves the sarcoplasmic reticulum. And once in the cytoplasm, it can bind to troponin. Troponin changes shape. Tropomyosin moves. And we get to the third part of this, the contraction phase of our skeletal muscle. And we do not have time today to do that. So we will do the contraction part on Friday.